Hi, good morning. My name is Mike Jewett, and I'm a professor in chemical and biological engineering at Northwestern University. And very excited to be here today to talk to you about some of the work that we're doing in the context of establishing cell-free biology for the production of therapeutics, materials, and chemicals. Of course, before I get started, I'd like to thank my team. I think just as in all research, the people that really do the work and really move the science forward, and we've also been very fortunate to be funded by a variety of organizations, and some of the work I'll be talking about today has been funded in particular by the Office of Naval Research, DARPA, and the Army Research Office. So very excited to have the support from those different organizations. Really what motivates me as a scientist is the idea that over the next century, food, energy, chemicals, and materials really, I believe, are going to be transformed by our ability to engineer biological systems, whether that being using a microorganism to, for example, produce pharmaceuticals or rubber for your tires, or even spider silk as next generation materials. And while I think we can all agree, certainly in the scientific community, about the importance of using biological systems to produce molecules as a powerful technology, the promised transformation is really moving slower than one desires. So here's just a snapshot of a variety of small molecule chemicals that have been produced heterologously over the last few years, at least at the commercial scale. So 1,3-propanediol, artemisinin, farnesene. And one of the things that really these systems have in common is that in order to engineer a microorganism to produce a small molecule, it's really costly and slow. So you can see the time to market there. In particular, in the decades of time here, 15 years to 13 years. And what's more noted is R&D years here. The time in which it takes these processes to be developed are in the hundreds of person years of time. So if I were to basically establish that for my own lab, that basically starts to run into the hundreds of PhD students if you're thinking about 500 person years. And so while engineering biology could be very useful, certainly contributing to the national bioeconomy and a variety of other formidable challenges that our society is facing, we'd like to step back and ask some questions about why is engineering biological systems so hard? And I think that there are many reasons, and certainly we're not the first to think of these, but there are a variety of possible reasons. So one of these is just that we have an incomplete knowledge of how biological systems work. So if I'd like to take a microorganism and feed it a sugar substrate and convert that into a valuable product, I might not necessarily understand all of the metabolic networks that go on inside of the cell, or the transcription factor regulation networks that I need to control in order to channel the ability of the organism to produce a product of interest for me. Secondly, cells have a lot of unforeseen interferences. There's unwieldy complexity, and oftentimes these are major challenges. One of the things my lab really centers on is that it's actually very difficult to balance intercellular fluxes to optimally satisfy active synthetic pathways while the machinery of the cell is functioning to maintain reproductive viability. So put another way, engineering living organisms is really hampered by our ability to manage both the cellular objectives, so basically evolutionary and adaptation objectives that a cell might have, but in addition, the engineer's process objectives. So basically, the idea that I would like to be able to synthesize a product. And this tug of war really is fundamental in our challenge to be able to use microorganisms as cellular factories. And so what comes to bear is that new tools are needed to harness the powerful and synthetic functional capabilities of biological systems. And I think that there are many exciting opportunities, particularly leveraging our newfound ability to read, write, and edit DNA. And there, within this context, what my lab really focuses on is something called cell-free systems. And really, these types of systems open the aperture of the traditional engineering model in biotechnology. So for example, what you're able to do is take a microorganism, which is shown here. You actually lyse that microorganism, and you harness the intracellular components of that microorganism essentially as a catalyst that enable you to convert raw materials into a variety of biobased products. And really what this affords you the ability to do for the first time is to truly decouple catalyst synthesis from catalyst utilization. And I think we're really excited about kind of this decoupling because this actually begins to separate now this tug of war that I mentioned as a fundamental challenge on the previous slide. And so this is really a new approach, it's an emerging approach, and I'd like to talk to you about some of the advantages and some of the new and exciting opportunities that are really coming from this approach. So in particular, cell-free biology, so this idea that we can activate complex biological processes without the cell. So basically what you've done is you've taken a cell, you've ripped off the cell wall, and you collect the guts. Think about it as kind of collecting the guts of the cell, or opening the hood, and now you can take that and you can direct that to produce a product of interest. The advantages of this are multifold. So first of all, you actually separate catalyst synthesis from growth, and so this is important because now all of a sudden we get to decouple the evolutionary objectives of the cell and those process objectives of the engineer. You lack structural boundaries, and this is actually subtle but remarkably important. It allows you for the first time to really have direct access to the reaction conditions that exist inside of the cell, and this is important because now you can not only do things that wouldn't be possible in a living organism, but you can additionally, so for example, you can sample the system of interest directly without being behind the barrier of a cell wall. You can also add non-natural components, and I think that there's some very exciting opportunities there. The other thing is that we lack cell viability constraints, so this allows you to perhaps produce molecules that would otherwise be toxic to a cell. It allows you to direct all of the resources of the substrate that you're feeding that you would like to be molecularly transformed into a useful product towards the exclusive production of that product. Again, we no longer have to also support life of the cell. And this really begins to offer a new paradigm of logistics and on-demand biomanufacturing. And so what I'd like to talk with you about kind of over the remaining 15 to 20 minutes of my talk are some exciting new avenues in this idea of biomanufacturing without walls. So the idea that you can utilize cellular lysates or cellular extracts to produce small molecules, protein therapeutics, and even materials on demand. So my lab actually works in three major areas. So the first area is in cell-free protein synthesis. So the idea is we use a crude lysate of a cell to produce proteins. And we'll talk about that for producing therapeutics and materials. We also are developing newfound abilities to basically create synthetic ribosomes. So the ribosome is the molecular machine of the cell that produces proteins, and we'll talk about that. And although we're not going to talk about it today, my lab is also working in the field of cell-free metabolic engineering for pathway debugging and biosynthesis of chemicals and materials. So let's talk about cell-free protein synthesis. This is actually a very historic field. So it was used initially in the 60s to discover the genetic code by Nuremberg and Mathai, and certainly others more recently. In order to carry out cell-free protein synthesis, what this means is basically I take the crude lysate of a cell and direct proteins to be synthesized. So I take, in this case, this is meant to represent the inside of the cell that contains all of the necessary factors for protein synthesis. So amino acyl tRNA synthetases, tRNAs, elongation factors, initiation factors, and ribosomes. And basically we grow up cells and we harvest those in mid-exponential phase. We lyse those cells and we prepare a crude extract. We then basically take that extract, we feed it substrates and salts. So in this case, the substrates and salts are necessary both for the product. So substrates are energy and such as ATP and amino acids are needed for producing proteins. And the salts are necessary to create the right physiological environment. We had a DNA template shown in kind of here, and that directs the synthesis of a particular protein. And so this is a combined transcription and translation system that exists inside of a test tube. So we've basically taken the fundamental molecular machinery of life and we put it in a test tube, and we're learning how to activate and control that. Really, there's been a driving force and several that have been driving the development of cell-free protein synthesis technologies. So first of all, these technologies are very efficient because they allow you to exclusively produce a single product. They can be simple, adaptable, and controllable. They allow you in particular to produce many, many proteins in large arrays. And they also allow for rapid product and process development timelines. So if you try to produce a protein, for example, inside of a cell, that often takes, at the current technologies, on the order of 1 to 2 weeks, depending on how fast this goes, certainly in one of my graduate students' hands. Whereas a cell-free translation system, once you have the plasmid or the DNA template of interest, it really takes on the order of hours to days. And really, advances in the last decade have made possible cost-effective high-yielding bacterial cell-free translation. So this is just an image which shows a protein being produced. In this case, it's chloramphenicol acetyltransferase on the y-axis. On the x-axis is time in hours. And so basically what we do is we add a cell-free protein synthesis reaction to the bottom of an Eppendorf tube. And then we watch the protein being synthesized with time. And so what you observe here, for example, is the amount of active protein being synthesized per time in the system. So we're producing up to about 700 micrograms per milliliter. And really, when you look even commercially, producing protein on about the order of gram per liter quantities is really significant. And importantly, basically, as the last decade we've been able to produce protein, in particular here we're showing about a tenfold improvement over an initial state of the art. It's really opened the way to new applications. So if you look over the last decade, you can see protein yield per time is basically increasing, where we're now producing on the order of multiple grams per liter in these test tube reactions. The synthesis rate has been increasing with time. The batch reaction duration has been increasing with time. And all of these combined with lowering of the overall economics, so the costs associated with the system, have even enabled microscale to manufacturing scale synthesis of these systems on the order of 100 liters. So there's really a dawn of a new era in cell-free protein synthesis systems. So you can see here 1996 in the microliter scale, and really we learned how to produce protein very cost effectively starting in about 2008 by activating respiration and glycolysis to drive cell-free protein synthesis. And these reactions are now being run at the 100-liter scale, which is just remarkable when you think about the development that's happened over the last 10 to 15 years. And so people are now trying to tap into that to see what and how to take advantage of these. There's additionally, beyond the bacterial systems I just mentioned, growing interest in developing simple, inexpensive, and high-yielding eukaryotic cell-free translation systems. So my lab, for example, is developing some systems in yeast, and we've shown improvements up into about the tens of micrograms per milliliter and are starting to move beyond that, and also significant reductions in cost, 1,000-fold improvement over where we initially started just a few years ago. Some other application areas, though, that I'd really like to delve into is this idea that we'd like to extend the central paradigm of biology to new chemistries and unnatural polymers. So one of the exciting things you might be able to do outside of the confines of a cell is to be able to expand the chemistry of life. And this is also being done in cells, to be clear. But cell-free systems offer some particular advantages for different unnatural amino acids that might otherwise be difficult to get across the cell membrane. So what you'd like to think about is to encode some protein or a polymer in DNA, allow for the transcription of that into RNA, allow for the translation of that into some protein or polymer by the ribosome. But we'd now like to, rather than use just the simple 20 amino acids that biology has provided us, or there are actually a few more than that, but the 20 canonical, we can then move to basically producing polymers of those unnatural amino acids, polymers with single unnaturals, with multiple identical, with multiple distinct, and all distinct. I think this is a vision certainly put forward by a variety in the field, among others, Peter Schultz, Jason Chin, and a variety of others that push this. But to achieve this vision, we need new tRNAs that can be amino acylated with non-standard amino acids, new codons that can be assigned to these amino acids, and ribosomes that allow for the formation of new polymers. And my lab is actually working on these in the context of cell-free systems. And really over the last decade or so, pioneering efforts have really demonstrated that it's possible to genetically encode more than 20 natural amino acids. And this is enabling some exciting opportunities, both genetic isolation for biosafety of different organisms, and the production of genetically encoded materials, new biological insights, and even designer protein therapeutics. So for example, the company Ambrex, also I know the company Sutro Biopharma is working in this space, among others. The problem, of course, with these types of systems is actually at the moment, they're not very efficient. So the state of the art for site-specific non-standard amino acid incorporation, where basically what you're doing is you're taking a tRNA and you're recoding it, in this case an amber tRNA, to leverage and use an unnatural amino acid that gets charged by a natural synthetase. This is then charged tRNA, is transported to the ribosome by EFTU, and this elongation factor TU ultimately drives the synthesis into the ribosome and competes against this release factor. So this is kind of the state of the art, how it's done, and amber stop codon suppression really uses 3 stop codons. More than more than 70 non-standard amino acids have been incorporated to date, but the average that's incorporated is about 1 to 3 non-standard amino acids per protein. Applications, however, remain severely restricted because of low efficiencies of incorporating these non-standard amino acids. And so what are the limitations? So first of all, the synthetases tend to have, that charge these unnatural amino acids tend to have poor catalytic efficiency, and that leads to sometimes inaccurate and inefficient incorporation of these non-standard amino acids. For some unnatural amino acids, the binding of this transport protein elongation factor TU is a challenge, and so you have to actually evolve this element of the translation apparatus. And lastly, for amber suppression, basically this amber tRNA is competing with a release factor that's endogenous in the cell. This causes early termination. And really over the last couple of years, this problem has been mostly solved by a variety of pioneering efforts, some shown here and others that we have actually been a part of. So in particular, Fair and Isaacs with George Church at Yale University and Harvard University have really created the first ever organism with a dedicated 21st codon. So they created a genomically recoded organism that basically lacks all occurrences of the amber codon and also is able to delete release factor 1. And so the publications are shown here. And basically what we've been able to do is that you can use these chassis organisms, so in particular these release factor 1 deletion organisms, to improve the synthesis of full-length proteins containing site-specific non-standard amino acids. So a publication for reference is shown here by Sekun Hong, who's one of my postdocs. It was recently published in ACS Synthetic Biology. The important thing is what we did is we compared protein synthesis with an unnatural amino acid in a release factor 1 present strain versus release factor 1 absent strain. And critically, what you observe, this is an autoradiogram, what you observe is the full-length protein of, in this case, a GFP protein that contains an amber at position 132, and here's a truncated protein. And when you lack, basically, or have, present release factor 1, what you see is when you try to produce the full-length protein, actually about 80% of your product shows up here in the truncated band. Whereas if you don't, for example, add the synthetase, you don't get any of the full-length product. We see a remarkable shift when you move to the release factor 1 absent strain. So in this case, very little truncated product. Most of the product is moved up to this full-length band. And that's very exciting for us. So you can see the quantitation of those numbers here. So wild-type GFP, in this case, we see the improvement in the absence of release factor 1 to this mark here. And so we're very excited about this, and I think that there are exciting opportunities to improve these systems. So this is based in a strain called REC13, where 13 of the essential genes and 7 of the essential genes and some others have been recoded with this amber. What my lab has then done is basically tried to design a better catalyst. So the idea is the cell lysate is a catalyst, and so you have a genomically recoded organism, and what we'd like to do is apply genome engineering strategies to improve the extract performance. So you imagine that if you change the overall composition of the catalyst or of this crude cell lysate, you could change the overall efficiency of the cell-free translation system. And so rather than using genetic engineering, which is kind of serial and has in some ways inefficient modification of the DNA with single to few genetic changes, what we're doing in my lab is applying genomic engineering, which allows for parallel site-specific efficient modification of the DNA, and using that to enhance positive and remove negative vectors. So essentially what we're doing is we're using this process called MAGE, which was developed in George Church's laboratory. We're then redesigning oligonucleotides, and basically each one of these bands on this gel show that you have a single protein that's been knocked out of the genome, both in individual molecules as well as a double. Here's a triple knockout. This is a quadruple knockout. And what we then did is we generated strains and E. coli lysates from these strains to produce products of interest. And in the end, the key result is shown here. So we have non-standard amino acid incorporation in these genomically recoded extracts, improves yields, and achieves high suppression efficiency. So what we observe in our work is that using a genomically recoded organism, we can produce, in this case, that only has 13 of the amber codons recoded, we can produce up to about 800 micrograms per milliliter of SFGFP with multiple unnatural amino acids in it, which really I think compares very favorably to to the state of the art. So basically you can see here, as compared to a variety of the other in vivo reports that are out there, in just a standard batch reaction, the in vitro system using the same unnatural amino acid has performed a little bit better. And so in summary, from this half of the talk, what I'd like to just kind of show you is that release factor 1 deletion strains can provide benefits for incorporating multiple non-standard amino acids site-specifically into the same protein, that genomically recoded organisms can be used to improve cell-free performance, and that looking forward, the development of release factor 1 deletion strains as a chassis for cell-free translation will open new avenues for using synthetic biology for synthetic chemistry. What I'd like to do at this stage is transition a little bit to one of the other stories that I mentioned we talk about, and that is in the context of synthetic ribosomes. So really the extraordinary synthetic capability of the ribosome has driven extensive efforts to engineer the translation apparatus. So not only, you know, and I show some statistics here, which is just that, for example, protein elongation rates in bacteria proceed at rates of 20 amino acids per second. They're remarkably accurate, and we do have genetic encodability. And so if you think broadly about the idea of constructing synthetic ribosomes, this was a part of the translation apparatus, but this could afford new opportunities for synthetic biology, both in minimal cell projects, in potentially driving virtual machines inside of organisms for orthogonal genetic control, and extending the central paradigm of biology to the complete synthesis of abiological polymers. So the unfortunate thing is that the requirement of cellular viability really constrains the mutations that can be made to the ribosome. So the natural ribosome must decode every RNA of the cell. Therefore, mutations in the cell to the ribosome are generally lethal, and they typically interfere with the endogenous proteome. So this advance has been in part addressed through something called the specialized ribosome system. But the issue is that you don't have the ability to engineer the large subunits of the ribosome. And so one of the goals that my lab has is to construct synthetic ribosomes in vitro. And the ribosome is really this large macromolecular machine, which comprises of about 54 proteins, so 21 proteins in the small subunit, which is shown here, 33 proteins in the large subunit, which is shown here. And we'd like to learn how to construct these in vitro. And so to construct these in vitro, we're trying to use a molecular stepwise approach whereby we both first can reconstitute a natural ribosome of completely in vivo purified parts. We then would have an in vitro synthesized parts that we can basically combine with the in vivo purified parts. And then we have, in this case, the ultimate construction of a synthetic ribosome. So this, of course, has been done before. So in fact, functional E. coli ribosomes have been reconstituted from their native parts. There are really some pioneering works by Traub and Nomura that were published in 1968, and Knut Nierhaus and Ferdinand Dome, which was published in 1974, showing the ability to actually reconstitute natural ribosomes. This reconstitution basically followed a kind of in vitro non-physiological approach, whereby you could essentially take a native large subunit, break it into its parts. So it's 23S ribosomal RNA, 5S ribosomal RNA, and the total protein of the 50S subunit. You then basically take all these parts, throw them into a test tube, and you can reconstitute this central molecular machine of life, which in and of its own is pretty remarkable. The challenge is that it wasn't very physiological, and the system was fairly inefficient. So in fact, if you look at major challenges in E. coli ribosome construction, ribosome reconstitutions historically have been non-physiological using these high salts and high temperatures. Ribosomes reconstituted with in vitro transcribed 23S ribosomal RNA were essentially non-functional. This is unique to the E. coli system. There are some thermophiles that have been used which were successful. And the coupling of ribosomal RNA synthesis and assembly have not yet been mimicked. So one of the things my lab did in collaboration with George Church at the Harvard Medical School is to create a novel platform for the construction of synthetic ribosomes, whereby basically you could synthesize in vitro the parts of the ribosome through transcription. You then allow the ribosome to assemble, and you allow for translation, all in the same pot. So basically you have an integrated synthesis, assembly, and translation system that occurs in a ribosome-free crude extract of cells, and you have one bacteriophage T7 RNA polymerase that transcribes both the ribosomal RNA and the messenger RNA of the cell. And so this system offered some interesting advantages for co-activating ribosomal RNA transcription and assembly, but it remained fairly efficient. So basically you have all these parts that you're adding into a test tube to allow for these ribosomes to be self-assembled and to be able to basically carry out their activity in protein synthesis, but they're very inefficient. So this is an E. coli ribosome, and this is the activity in the context of producing a protein of these synthetically constructed ribosomes. So they're actually about a little more than 10,000 times less active than a purified E. coli ribosome. And so we hypothesize that the activity of this synthetic ribosome construction system can be increased by several approaches. And several of these are published now. So one is in the nucleic acids research that my student Brian Fritz recently published. Liu has published something in ACS synthetic biology. And really the end result is that over the last 4 years, we've been able to increase activity of this synthetic ribosome construction system about 10,000 fold. So you see here, basically activity is a measure of the amount of protein we can produce, and you can see this large increase over time. And we're now almost as good as a natural E. coli ribosome in our system. We've also established that this platform can make modified ribosomes, and that's very exciting because this will be important for exploring ribosomal mutants that potentially might also be used for incorporating unnatural amino acids, like we talked about in the earlier part of the talk. So what we show in this figure is luciferase production on the y-axis. We use 2 different ribosomal RNAs, one that has a resistance mutation, a mutation that makes it resistant to the antibiotic clindamycin. And what we see is that in the presence of clindamycin, only the ribosomal RNA that is resistant to that antibiotic actually is functional. And so this was a very exciting and important proof of concept. And so what we've been able to show is that ribosomal RNA synthesis, assembly, and protein synthesis can be co-activated without living cells, and that ISAT, this integrated synthesis, assembly, and translation system, can enable the rapid construction of synthetic modified ribosomes in vitro. Looking forward, I think we anticipate that ISAT will aid in the variety of studies in ribosome assembly and increasing physiologically relevant conditions and open new avenues for ribosome engineering. So I've tried to talk about a couple of vignettes today. One focused on the ability to actually carry out cell-free protein synthesis and release factor 1 deficient strains, and I've shown that this allows us to really incorporate unnatural amino acids at a fairly unprecedented scale, at least of the same type and at very high yields. I then went on to show that basically we could carry out in vitro synthetic ribosome construction in these systems. Cell-free biology ultimately, I think, is going to offer some new frontiers for advanced bioproduction systems, both because it reconceptualizes the way we engineer biological systems by truly really decoupling this idea of catalyst synthesis from catalyst utilization. There are definitely some key challenges that the field is working on, so scale up, efficiency, moving beyond kind of E. coli, which is the common organism that's used in the field. But I think we're going to continue to see these developments over the next few years. So there are certainly some new frontiers in cell-free bioproduction systems, as we talked about, design of ribosomes for the production of new types of polymers and materials, rapid on-demand biomanufacturing for antibodies and vaccines, and sustainable synthesis of commodity chemicals and materials I didn't talk about. But I think that the future is certainly very bright. So in closing, I would just like to acknowledge a variety of the people that did the work. So in particular, the work that I showed today. Sekun Hong worked on the release factor 1 mutation strains. Brian Fritz, shown here, and Liu, shown here, have done a variety of the work in the in vitro ribosome construction system. As I mentioned, our lab has been sponsored by a variety of organizations, and it's really important to have those organizations supporting us, such as, for example, the Office of Naval Research, DARPA, the Packard Foundation, the Army Research Office, and the NSF, which all contributed in some way to some of the research that you saw here today. I'm also very fortunate to have a variety of collaborators that we work with in a day-to-day basis, and so that's very important. Finally, I'll close with a slide from the lab showing us here in Chicago. We're north of Chicago and Evanston. This is about the view from where our lab sits. And I'd be happy to take any questions that you have at this time. Please feel free to type them in the box if there are any that come to mind, and I really appreciate your time and attention today. All right, so one of the questions that just came in is the following, and I'll try to cover this briefly. But basically, it's asked, how durable are cellular components after they're removed from the cellular environment? So once you break the cell wall, can they be continuously used as long as substrates are provided, or do they lose efficiency very quickly? This is a really important question. And I'm actually going to go back to one slide to show the batch reaction duration. So if you see here on this, excuse me, I just want to be able to get the pointer. So if you see here, basically batch reaction duration. So here we have this lysate of a cell that is active in a batch reaction producing protein for up to 12 hours. So that gives some indication that for at least 12 hours or so, these proteins, without actually adding any extra substrates or anything else, can remain active. We've actually observed in some cell-free metabolic conversions the ability to move beyond 24 hours and still maintain activity. Some of the longest reports in the literature show that as long as you're continuously feeding substrates and continuously removing any byproducts, then you can actually get these reactions to run for days. It seems that I think the longest in the literature is about 5 days. Vincent Nouriot, who's at the University of Minnesota, is one of the key people in the field who's been able to show some of this work. Of course, Alexander Spirin was the original, showed the original work that that's actually possible. So I think it's very exciting, certainly that you can activate these systems outside the cell and they remain active. In terms of the extent of to when the system actually breaks down, we don't know the answer to that question. So thank you for that question. I appreciate it. I'm available for other queries if there are any. So we've actually had another couple questions roll in. I'll try to answer at least one of them here. Again, so one of the questions is, do proteins fold efficiently outside of the cellular environment? So if you take a protein and so let's say I want to produce protein X, how efficiently does it fold? So interestingly enough, this is one of the exciting opportunities of the cell-free translation systems, is that proteins tend to fold fairly efficiently. And it's actually for two main reasons. So one of the main reasons is that so for example, let's take a bacterial cell-free translation system. We produce molecules anywhere from full-length antibodies to tissue plasminogen activator, which is a molecule that dissolves blood clots and heart attack victims, to granulocyte colony stimulating factor, which helps kind of stimulate white blood cell production, to even single-chain antibodies. So we can produce molecules with complex disulfide bonds, and even things that need folding partners. One of the things that seems to be important is that the cell-free translation systems are an order of magnitude slower in translation rate than what occurs in cells. So in the context of the bacterial cell, protein synthesis rates are on the order of about 10 to 15 amino acids per second. Whereas in the cell-free translation system, we tend to observe on the order of 1 amino acid per second. And what that means is that ultimately the slower translation rate tends to help proteins fold a little bit more efficiently than they would otherwise inside of a cell. So the second reason why folding is oftentimes advantageous in these cellular lysates is actually because of the dilution effect. So the cellular milieu is on the order of 200 to 300 grams per liter of protein concentration, whereas these cell-free lysates are on the order of 10 grams per liter or so. And so that reduction of about 20-fold ultimately means that as proteins are being folded, they have a lower chance to actually aggregate. So if you think of protein folding as a unimolecular event, so a protein comes off the ribosome and it folds, protein aggregation is actually a dual effect. So basically you have two proteins that come together to begin aggregating. So it's always a second order effect or greater. And as you think about that, basically the magic rule in protein refolding is to keep solutions dilute. And so it just happens to be that we actually benefit to produce proteins in their active conformation by using a lower concentration, or actually the cellular concentration of just kind of general proteins is lower. So another question that's popped in that we should talk about, and please feel free to keep asking questions. We'll see how many we can get to. Is actually the question of how different is the synthesis of therapeutics from chemicals in vitro. So in the context of cell-free protein synthesis systems, I should add that basically the state of the art at the moment is we can begin to produce complex therapeutics such as full-length antibodies. But there is not yet, for example, a Food and Drug Administration or an FDA-approved process for producing protein therapeutics in vitro. I think that's going to change in the next few years. But essentially, that process is producing a protein. And so when you do that, what you need to add to the system is a DNA molecule that encodes for, let me actually go back to the slides here. But basically, you need to add a DNA molecule that encodes for the protein of interest you'd like to make, and that allows you to basically do that. The trick is with chemicals, it's a little different. So if you want to think about producing chemicals, you're not actually overexpressing, generally, proteins of that process, at least in a crude extract system. In that case, what you're actually doing is you are simply trying to provide the catalysts that allow for conversion of, let's say, glucose to whatever product you have of interest. And pretty at least in the context of crude extract systems, Sven Penke's lab at the ETH is one of the more predominant ones. Jim Schwartz's lab at Stanford has really shown some beautiful work, as well as his company, Greenlight Biosciences. In the context of purely in vitro systems. So all the systems that I talked about today were crude extract systems. There are a lot of works out there in the context of chemicals, and most of those chemical systems are actually done with purified in vitro systems. So there are actually commercial processes with single purified enzymes, so glucose isomerase, for example, which helps make fructose and others. But there are very few commercial examples of multi-enzyme pathways, but that is a very active area of research. It'll be interesting and exciting to see what opportunities come about in the future. Another question that's popped up here is what kind of challenges do you envision for the scale-up of your cell-free system? So really there are a couple. The key thing is the substrate cost. So one of the things that had always historically limited cell-free translation systems, for example, to small scale, were the costs. So if you look at this figure here, where we basically have small reaction scales, so reaction volume on liter scales. So we're down here in the microliter scale for such a long time. Basically, in this case, what we actually are observing is that these systems. Excuse me for 1 second. These systems are so small because of the dominant cost here, really the energy substrate. So you would end up having, for example, phosphoenol pyruvate as a secondary energy substrate, which is about $160 a gram. And so you can compare that to, for example, glucose, which you could buy from Sigma for 5 cents a gram or something. And so you can see that really the important element is basically to feed a cell-free system the same type of molecule you would feed a cell. That's one of the core constraints. The other thing is kind of just cofactor and energy regeneration, so turnover numbers. Cells are very good at that, so how do we recapitulate those processes? And the third, really, I would say, in the context of challenges for scale-up, is this idea that we'd like to keep the proteins active. So there's not really a way to regenerate a protein, at least on a continuous basis. So I think that there are some process engineering objectives and opportunities, kind of classical bioprocess engineering, that could be applied to these systems in the future. We could also begin, because the systems don't have walls, you can begin to open yourself to the decades of work that's gone on in reaction engineering by, for example, heterolysis catalysis, by kind of classic chemical engineering methods, and use standard kind of online detection and control, I think, could be applied to these systems. So another question that's come up is, can you comment on the prospects for using cell-free systems as prototyping environment for developing pathways to be scaled in cells? So this is a great question, and actually we're doing this now in my lab, and would be happy if you reached out to me post this conversation to talk a little bit more specifically about that, Jim, I think that there's a significant development right now, both using purified enzyme systems as well as extracts to achieve that exact goal. So there have been a couple publications in vitro purified systems, which have just shown the ability to prototype and identify the right ratios of enzymes. And there are also some opportunities in extracts. But I think that that's definitely a direction that the field is moving. Are there other questions that people have? I've gotten through the bulk of the list here. I'd be happy to take a few more. Okay, there don't seem to be any other questions rolling in, so I'll wait a few more seconds here if you have something that's burning. Otherwise, for those of you that tuned in, it was actually a little interesting to try to present this way without actually being able to see the audience. But please do feel free to email or follow up with any questions that you may have to me. I'm sure you can find my email online. Mention that you had seen this presentation, and I'd be delighted to talk with you more fully or in a different environment if you would like to do so. So with that, thank you, I guess, for all of your great questions. I hope everyone enjoyed the seminar and enjoy your day.